Hi, everyone. Welcome to the Cerebellum seminars. Today, as our guest, we have Aparna Suvrathan. She's an assistant professor at the Center for Research in Neuroscience at McGill University in Montreal, and she's agreed to give us a talk today on the different roles of plasticity and the timescales of them. She's affiliated with the Department of Neurology and Neurosurgery and Pediatrics at McGill. And I'll tell you a little bit about her background. She received her PhD at the National Center for Biological Sciences in Bangalore, India, a place that I've visited. And I can tell you it's an amazing place because you get to see macaques in their natural environment. And you also get to see the absolute respect for the environment that I hadn't appreciated until I traveled to India. Anyway, she did her PhD under the supervision of Sumantra Chatterjee, where she investigated the role of amygdala in a disease called Fragile X. Then she came to the United States. She went to Stanford University, and she was a postdoc at the laboratory of Jennifer Raymond, where she did really groundbreaking work in trying to understand the timescales of plasticity of Purkinje cells, and that's what she's going to tell us more about today.
Thank you so much, Reza, for inviting me and for giving me this chance to tell you all about some of the work we're doing today. I will start by talking about some of the work that I started in Jennifer's lab, which is to do with timing, but timing for plasticity. But I'm going to talk a little bit about synaptic timing more generally in work that is coming out of my lab. Actually, it was published a couple of months ago. And we will hopefully build this story about heterogeneity and how this affects cerebellar processing. Sort of to give an overview, the overarching goal of my lab's research is really to try and understand how the properties of synapses and cells, they lead to and support behavior. And of course, one aspect of this is learning. So I will start with talking a little bit about plasticity rules, but just a few slides because this is now a very old study. And then I will try to move on to a more recent project from my own lab, which talks about a different aspect of synaptic timing, but still really emphasizes this idea that we need to consider heterogeneity in synaptic function and its consequences for the cerebellar circuit. So to this cerebellar audience, I really don't need to talk about the fact that when we talk about plasticity rules, of course, one of the major frameworks in terms of which we think is Hebbian types of learning rules. And more specifically, we often talk about plasticity rules. Let me see if I can have a pointer here. Do you see my laser pointer? Does that work? Okay, perfect. We tend to talk about plasticity rules that function on the time scale of milliseconds, where synapses are strengthened or weakened depending on their order and their close to coincident occurrence. However, many signals that guide plasticity in vivo, when we move away from this word that us synaptic physiologists tend to think about, the plasticity in the slice preparation, but you consider how this plasticity occurs in vivo, signals that guide plasticity, be they reward, punishment, or in the case I will talk about, error, often come back to signal to the circuit that something needs to change at much longer time intervals, ranging from hundreds of milliseconds to seconds. Now, this confound, of course, has several potential mechanisms by which you can resolve this disconnect between millisecond scale plasticity rules and the requirement for longer time intervals during behavior. But when I was in Jennifer's lab, we found one mechanism, one way in which the cerebellar circuit solves this problem, and that involves addressing the temporal credit assignment problem at the level of synapses. And let me explain that a little bit more. So in this hypothetical model where we have Purkinje cells, and in this case, we're going to talk about a specific region of the cerebellum called the flocculus. The cerebellum drives behavior. If that behavior is erroneous, so there's an error in it somewhere, that error is signaled back to the Purkinje cells. We think, and of course there are different models about this, but one of the dominant models is that the climbing fibers signal this error back to the Purkinje cell. And now the temporal disconnect here is that when we as plasticity people study forms of plasticity, and more specifically forms of long-term depression that we think underlie learning, we use protocols where it is important that parallel fibers and climbing fibers are coincidentally active. And yet, for this loop to occur during behavioral learning, there has to be a delay in time before that climbing fiber error signal goes back to the circuit. Fortunately, because the cerebellar flocculus and the behaviors, the oculomotor behaviors that it supports have been very well studied, we have an idea what this delay is. And therefore, we could plug that delay into the floccular circuit. So as I mentioned, I don't want to spend too much time on this story because it's really many years old right now. But what we found was that classic forms of plasticity induction, where you drive parallel fiber and climbing fiber activity coincidentally, are not effective at driving plasticity, specifically depression, at the Purkinje cell. However, if one were to delay the climbing fiber, the CF over here, by 120 milliseconds, that specific delay that it takes for the climbing fiber error signal to come back to the floccular circuit, you are then able to drive depression at these synapses. And that's sort of summarized here, where 0 is the coincident application of parallel fiber and climbing fiber. There is no depression. In fact, there's potentiation. But at 120 milliseconds, you do get depression of these synapses. And therefore, what we found was that long-term plasticity in the flocculus is accounting for this time delay for the error signal to come back, which is rather nice, because we tend to think of our synaptic plasticity and our synaptic plasticity rules as independent of sort of circuit function. We sort of invoke the same plasticity rules, but we apply them to different circuits in a universal manner that we suggest is not entirely true. Now, this form of plasticity induction for the plasticity aficionados was true across a range of conditions, accounting for the fact that parallel fibers fire in bursts, where they do it in current clamp and voltage clamp. The point to notice is that if you delay the climbing fiber by 120 milliseconds, you get effective long-term depression at parallel fiber synapses, which you do not do when this is coincident. And of course, what you're all wondering is just how precise is this? It's fairly precise. So if you had this on the x-axis, the interval between the parallel and the climbing fiber, or the climbing fiber delay, you can see that plasticity is quite narrowly tuned within a few tens of milliseconds to a delay of 120 milliseconds for the cells in this circuit. And just that's kind of on the order of spike timing dependent plasticity rules, just that they are tuned to the delay of the climbing fiber for this circuit and for the functions supported by this circuit. So in addition to looking at this at the synaptic level, we also considered whether this would be true in vivo, because after all, when we make a slice and we block inhibition and we have all these inputs cut, we are essentially working with a very artificial preparation. We did a few things to try and address those confounds, including moving away from protocols for long-term plasticity to protocols for single trial plasticity, which is similar to what happens during motor learning in vivo. I will not talk about that today because you can go check this out. But what I do want to mention briefly is that in collaboration with Hannah Payne in the lab, when we looked at whether these kinds of plasticity rules occur in vivo, whether there's any evidence that this is not just a phenomenon that one observes under the artificial conditions of an ex vivo preparation, but whether in the intact circuit during actual learning, there is evidence that there are timing rules for plasticity, we consider that if one looks at trial by trial learning during vestibular ocular reflex learning, which is the form of ocular motor learning that the flocculus supports, which this audience, I'm sure, is very familiar with, then from one trial to the next, if there is no climbing fiber activity on the first trial, there's no change in Purkinje cell firing on the next trial. And it has been well established that if there is a climbing fiber firing and there's a complex spike coming in, then you do have a small depression of the Purkinje cell on the very next trial. Now, those data that suggested there's this trial over trial learning were, unsurprisingly, always aligned to the trial structure of the behavior that was being studied. Whereas what our data suggest and what we are suggesting is that temporal precision is relative to the time of the climbing fiber for that cell for that trial. So what we did was mine data that already existed of monkeys, this is from rhesus macaques, doing vestibular ocular reflex learning. And what is different now is that your x-axis is the time from the climbing fiber spike on the previous trial. So that's aligned to 0. So that's really the only difference from other trial by trial learning data. But when you do this realignment, you find that the depression on the next trial is quite nicely timed about back in time 120-ish milliseconds from the time of the climbing fiber spike, which would happen if it drove the plasticity of synapses active before it. This precisely timed in vivo plasticity was paralleled by slightly later an adaptive change in eye movement, which is also, precisely timed in the direction we think. And so to just sort of wrap up this part of it, what we think is happening here is that if you had a hypothetical Purkinje cell, which had hypothetical parallel fiber synapses, sets of synapses, say 1 and 2, and on trial 1, there was an error caused by one set of synapses or the activity of those synapses, that error is not signaled back via the climbing fiber for a delay delta t. And that delay is how long it takes for the climbing fiber to come back and signal to all the synapses on the Purkinje cell that there was an error. So depression is now driven not at the synapses that are actively coincidentally with that climbing fiber, because of course these are not the synapses that drove the error, but they are occurring at the synapses that are active exactly back in time, delta t, which for the flocculus is 120 milliseconds, thereby accounting for that delay and also ensuring that it is the correct synapses that undergo depression. So it's sort of solving temporal credit assignment at a synaptic level. This went on we sort of have this working model based on work that we did, recordings that we did outside of the flocculus, that while in the flocculus there is one kind of error signal, which takes 120 milliseconds to come back, and therefore plasticity is tuned to this window of 120 milliseconds. There are other regions of the cerebellum that encode errors with different delays. And this is more a working model than proof, because the modality of the error is often very hard to map on to place in the mouse. However, what we do observe is that in the vermis, in lobules that support several different functions, ranging from simple motor behaviors to cognitive functions, there is a range of delays to which Purkinje cells are tuned. And so we think that there might be, and this is our current working model, there might be these multiple detectors that are tuned to different plasticity intervals and thereby help solve this problem of delayed error signals in the cerebellum. So just to sort of summarize without showing you all the data, what we found was that there were different rules for plasticity at parallel fiber synapses across the cerebellum, that there were precise timing requirements, and this was true long-term as I showed you, short-term as I did not show you, in vitro and in vivo, and that plasticity is essentially tuned to a delay that is relevant to the local circuit and the behaviors that the local circuit supports, in a way that, would provide a synaptic solution to temporal credit assignment, part of the temporal credit assignment problem. And ultimately, there were a diversity of these temporal learning rules across the cerebellum. So this study, which is from several years ago now, ties in to a more recent set of studies which have come out in regions such as the hippocampus, the amygdala, the mushroom body in insects, which all suggest that we need to move beyond the idea of only coincidence or close to coincidence based learning rules for plasticity, and that we need to account more fully for the function of the circuit when we consider the kinds of plasticity rules that are in play. Some of these studies came out a couple of years after ours, and they're absolutely fascinating because the time scales of plasticity have been driven out. We thought we had pushed it far beyond coincidence by going to 120 milliseconds. Some of them have gone as far as 2000 milliseconds, which when it's relevant for that particular behavior. So we think we sort of fit in with this larger move towards understanding plasticity rules in a more in a way where you account more for the function of the system. Before I get to the second project that I really wanted to spend most of the time talking about, I want to briefly mention one direction that this project took, the plasticity rules project. And again, I will not spend too much time on it because it's up on bioRxiv, and it was taken forward by a very talented postdoc in Jennifer Raymond's lab, Sriram Jayabal. But at the time we found this plasticity rule, we really wondered, why is that? How do cerebellar floccular Purkinje cells know that they should undergo plasticity at 120 milliseconds? What makes Purkinje cells in the flocculus different from Purkinje cells elsewhere? Why should it be tuned to 120 milliseconds? And one part of that question is, of course, whether this comes about through experience. Is it that Purkinje cells learn that 120 milliseconds, or is it that they're somehow hardwired? This would be an interesting answer either way. So what we did was rather crude in this circuit where the error signal, the error for the behavior is retinal slip, and that is signaled back by our climbing fibers. We sort of did this hammer on the head approach where we dark reared the mice. And that would prevent any visual input throughout development. And we wondered whether that timing rule would still be in play. And this is now data from Sriram. I'm just going to show you one figure where if you had coincident parallel fiber climbing fiber activity in the normally reared mouse, there is no change in Purkinje cell firing rate. In the dark reared mouse, coincidence actually does give you LTD. In the normal mouse, 120 gives you LTD, but in the dark reared mouse, 120 does not. So I will leave you there because the rest of this work is really beautiful and it is now mine. But I think that is really helped us think about how these plasticity rules actually come about. And there's some beautiful data there on how this is even tunable outside of development, that even in the adult, this can be tuned. So what I'd like to tell you about now is the more general question of, with these beautiful cells in this beautiful circuit, are synapses actually uniform? We found that in terms of plasticity, they aren't, in terms of plasticity rules. But more generally, the cerebellum, as we now know, is supporting a diversity of functions, right? You've got the flocculus doing very simple reflexive adaptation, but you've got social involvement, you've got cognitive behaviors, you've got all this complexity across the different lobules of the cerebellum. We've moved beyond the idea of, a universal cerebellar computation, but we do still think in terms of the same kind of circuit everywhere. And we wanted to interrogate this idea a little more thoroughly in my lab. So we know that there is a lot of molecular heterogeneity. we don't necessarily understand how this links to synaptic function and how that then bridges to the function of the circuit in terms of Purkinje cell output. So we looked at synaptic transmission just at parallel fiber synapses and asked the question, is synaptic transmission at longer timescales, at behaviorally relevant timescales, is that indeed uniform? So within at parallel fiber synapses, as I'm sure you're all aware, there is the fast AMPA receptor dependent current, which is, as far as we know, fairly uniform across the cerebellum. But we were interested in another current that comes through at the parallel fiber synapse, which is a slow cation current, which acts on a timescale of hundreds of milliseconds. So this slow cation current is dependent on metabotropic signaling. So it really comes because glutamate binds to the metabotropic glutamate receptor, and it is a non-specific cation current that comes through the TRPC3 channel. And I'll go more into this pathway a little bit later. But this is what this slow cation current depends on. And so I'm going to tell you a little bit about a project by Ria Thomas in my lab. And Ria actually just defended her thesis last week. So we're all very proud of her. And what she found was that when she looked at the slow cation current across regions of the cerebellum, and she looked at our old friend the flocculus, but she also looked at the midline vermis, the region most of us study as synaptic physiologists, and specifically looked at different lobules in the vermis. And when she did so, she found that this slow cation current, it's indeed slow across all regions, but that's a 500 millisecond scale bar, but it is also highly variant across regions. And that in lobules 4-5 of the vermis, you had a much faster current than you did in the flocculus or in lobule 10 of the vermis. The reason we chose these lobules is not entirely arbitrary, but bear with me for a little bit, but the reasons will become clear. So what Ria found was that these slow currents are actually surprisingly heterogeneous. The amplitude of the currents was reasonably similar. There was a slight reduction in lobule 10, which is consistent with previous work, actually. But what was really noteworthy was that this time of peak, the time at which the current peaked, was highly different between the three regions. What's also noteworthy, and this is tens of cells all smooshed up. What's also noteworthy is that there's a lot more variability in these two lobules of the cerebellum than in lobules 4-5. So you have a fairly homogeneous population here in the vermis, and in lobule 10 also in the vermis, and in the flocculus out in the hemisphere, you have much longer delays for this current, as well as more variability within those regions, and that's reflected by the coefficient of variation. There are different properties of this current that are noteworthy. It's not just the time of peak, it's the decay time, which shifts in a corresponding manner, the half width. And if one considers the amplitude of each of these currents versus the time of peak, which of course are related to each other, then one notices that lobules 4-5, regardless of the amplitude of this slow synaptic current, you have a narrow range of time of peak. So it's really tightly distributed. Less than 200 milliseconds shifted. However, in the flocculus, you have a completely non-overlapping population where you have much delayed time of peak, and lobule 10 actually falls somewhere in between these two populations, right? So it's really diverse. You have one tight population, and then you have these two lobules with all this variability. And of course, we wondered what this is all about. Where is this variability coming from? And that comes to a little bit about why we chose these regions. Now, as of course everyone in the cerebellum knows, the cerebellum is patterned into Zebron zones, which are seen better in the coronal section. But I just wanted to highlight that lobules 4-5 is a largely Zebron negative region, while lobule 10 is a largely Zebron positive region. So this coronal viewpoint shows you the sagittal striping of these Zebron zones more clearly. But we started by considering vermis and flocculus as regions where we knew there was potentially differences in timing, differences in function. And then we worried because in addition to difference between vermis and flocculus, lobule 4-5 is largely zebrin negative, whereas the flocculus is largely zebrin positive. So we wanted to pick a region that is now in the vermis, but is in the posterior, like the flocculus, and is zebrin positive, and therefore lobule 10. And so this is where we now get sort of the vermal hemisphere anterior posterior zebrin non-zebrin division between the regions. So what Ria then did was say, okay, we're sort of, if one simplifies this finding entirely and says, without going into too much detail of the shape of it, we could say in terms of timing, we have a short delay region in 4-5, and we have two long delay regions. Is it just that the two long delay regions are zebrin positive and the short delay region is zebrin negative? That would actually be consistent with our data. And as you know, there are many molecules that co-vary with these zebrin stripes, including many components of the mGluR signaling pathway. So it would not be unreasonable that, this is just, I say just, but is it just that it is in a zebrin zone that is positive, and those will have long delays, and the ones which are negative will have short delays? So to resolve this question, what Ria did was she capitalized on the fact that every zebrin negative zone, not every zebrin, but the zebrin negative zone in 4-5 has a few zebrin positive cells in between. There are narrow bands of positivity. And in the positive zones, there are a few negative cells. And so this natural variability, it helped us try and understand whether it was the zebrin identity of the cell that was determining what its synaptic timing was, or whether it was its location within a lobule that determined its lobular, whether it was its lobular identity that determined it. So what Ria did is, and here this is just staining to show you that lobules 4-5 is largely zebrin negative, whereas the other two regions are largely zebrin positive. So what she did was this fairly brave experiment where when she recorded the synaptic current from a cell, she dye-filled the Purkinje cell, and then she sort of post-fixed and labeled for zebrin so that she would then be able to see, and this is the dye-filled cell, and you can see that in some cases these are zebrin positive, and in some cases these are zebrin negative cells. And so she's now able to separate these two populations. So to summarize what she found, in lobules 4-5, black is a zebrin negative cell, and a green dot is a zebrin positive cell. So the zebrin negative population is the majority in lobules 4-5. This is exactly as we'd expect. However, there are a few zebrin positive cells in between, but regardless of whether they're positive or they're negative, as you can see, they cluster in this shorter time delay period. In contrast to that, if they are in the flocculus or lobule 10, they are much more likely to be zebrin positive, which is what we expect. But there are a few zebrin negative cells, which we also expect. And those zebrin negative cells, those few zebrin negative cells, still follow, they sort of sit within the population, right? So they're not separated based on their zebrin identity. And so what these data from Ria suggested to us was that while there is a broad correlation with zebrin identity, because these lobules are largely zebrin negative or largely zebrin positive, the zebrin identity of a Purkinje cell is not predictive of its timing. So we cannot just by saying, okay, this is a zebrin positive cell, it's going to have a long delay, and this is a zebrin negative cell, it's going to have a short delay. That did not work. So a zebrin positive cell in lobule 4-5 was still tightly timed to the shorter delay. It's still a short delay cell. So this was exciting to us because it sort of disproved one of the things that we thought was likely true, that it was dependent on zebrin identity. And it suggested that there's there's something more going on within these lobules, which overrides just the presence of zebrain. And I will come to that bit of the molecular aspect of it in a few minutes. But the next thing we worried about was that in order to get these slow synaptic currents, essentially what one does is one has to activate the metabotropic glutamate receptor. Excuse me. So to activate mGluRs, you actually give a train of parallel fiber stimuli. So we're blocking AMPA receptors, we're giving a train of parallel fiber stimuli, and then we're seeing the slow synaptic current. And we wondered whether this observation of heterogeneity was limited by the specific protocols that we used. And we'd used what is standard in the field, 10 parallel fiber stimuli at 100 hertz. And, we see the slow synaptic current. However, in vivo, parallel fibers are known to fire at that range, 10 at 100 hertz, but also a fewer numbers. So I think the lowest that's been observed is 3 parallel fiber stimuli together. So sort of 3 granule cells firing. A granule cell firing 3 times. All the way, and the frequency can go all the way up to over 200 hertz. So we wondered whether this was a general principle, this heterogeneity, or it was somehow restricted to the specific parameters that we were investigating. So what Ria did was first, she looked at different numbers of stimuli across the regions, and I'm just going to show you part of the data. The amplitude is the same, but the time of peak and the decay time varies exactly as it did previously. I know these figures are tiny, but essentially it's just to show that your 4-5 is still got an early time of peak, and your flocculus and lobule 10 have a delayed time of peak. So across a number of stimuli, this was true, and across a range of frequencies as well, this was true, that you still had the same, tight time of peak for 4-5 and the more delayed time of peak for lobule 10 and the flocculus. And so we were fairly convinced that this is not just an artifact or a very specific to 10 at 100, which would really limit its relevance. So across this range of stimulus frequencies and numbers that we think, as far as the literature suggests, occurs in vivo, this is highly relevant. So next we came to the question, which I'm sure all of you are asking. So what is this? Why are these cells different? Why are these currents different? And so we started by going back to what we knew about these currents, which is that they are mGluR and TRPC3 dependent. And we wondered, is that actually indeed true across all lobules? Because people, when they do these experiments, don't specify which lobule it is. So first of all, is it the same pathway everywhere? And we did this sort of classically. We blocked with an mGluR blocker, a non-specific TRPC blocker. It's really just a tyrosine kinase inhibitor, a more specific TRPC blocker. And in all cases, the degree of blocking was as expected from previous literature and was not different between lobules. So all 3 are equally dependent on mGluR and TRPC. We wondered whether it was the excitatory amino acid transporters, which of course would shift the timing of the current. It is not that. It is not sufficient to explain the shift in current. So we come back to this pathway that I showed you a little bit earlier. Where is this heterogeneity in timing coming from? And it's quite a problem, as you can imagine, right? We've got the simple circuit which says mGluR and mGluR signaling leads eventually to activation of this TRPC3 current. But there's a lot of other things in play over here, some of which are still quite controversial, about the involvement of various other candidate pathway components that everything that I've put in this diagram, there's some evidence that they either regulate or directly control current through the TRPC3 channel, including GABA B receptors, including the orphan receptor GLUD2, right? So depending on what system it is, and like I said, some papers contradict each other, but this would be everything that we think could play a role in regulating the timing of the TRPC3 current. And of course, manipulating each one of them individually is messy because others compensate. There are coupling effects that are sort of not easily separable. So what we did then, this was about the time, this was about 2021, and at that time, this really nice paper came out, which had RNA-seq, had a large RNA-seq data set, which was publicly available. So really, thank you to this group who had made this available, where they suggested that if you sort of high dimensionally cluster Purkinje cells based on their transcriptome, you have 9 different classes of Purkinje cells. Now, ALDOC is aldolase, which is zebrin. So there are 7 zebrin positive clusters and 2 anti-ALDOC or zebrin negative clusters. So that's zebrin positive and negative. And that this is sort of in high dimensional transcript space, the way Purkinje cells fall, that they fall into these 9 clusters. So given this data, we said, okay, this is wonderful, because now we can take their data, and they beautifully had lobular identity in there for which lobule each one of these are all single nucleus studies, each one of their nuclei came from. So what we did then was mine that data set for everything that is in this pathway, right? So exactly for those components. And that's what we came up with. So these are the mRNA for each of these proteins. These are the lobules. The ones I've highlighted here in color are the ones we're interested in, 4, 5, flocculus, and 10. And it's super confusing. So let me just highlight a couple of transcripts of interest and tell you why we're excited about it. First off, does it match what we know? So TRPC3 itself is significantly higher in 4-5 than in the flocculus and lobule 10. This matches with data that is previously known about zebrin identity and the lobule identity. So wonderful, sanity check. This works really well. The metabotropic glutamate receptor itself, GRIM1, is also higher in 4-5 versus the flocculus in 10. Something we also know based on previous literature and on data that I will show you that we experimentally tested. However, what we did not know, what is really exciting, and I'm going to highlight 2 or 3. Let's just see where it is. KCG is protein kinase C gamma, which has this inhibitory effect on TRPC3. And that is much higher in the short delay region than in the long delay regions, which would be consistent with cutting off the current early. So that would be great if that was our one candidate. However, several other molecules went in the same direction. So this is now DGKG, which is diacylglycerol kinase. So it sort of acts on diacylglycerol, and diacylglycerol has a role in activating TRPC3. So if you had more of it, you would lessen the impact on TRPC3. And indeed, the short delay region has much more of the kinase than the other 2 regions, which is what you would expect that kinase to do if it has to shorten the activity in that direction. We also saw STIM1 is higher, and this role of STIM1 is actually very, it's not accepted by everyone in the field, but it does have a regulatory role on TRPC3, and it has it in the direction we see here. That's also true of the data on the glutamate receptor delta 2. So we sort of ended up with a superfluity of riches. We were hoping that we'd find one that would explain that one is shorter than and the others are longer, but it ended up that several of these pathway components, all of which interact with each other in complex ways that are not fully understood, go in a direction that, based on what we do know, would shorten or elongate the current. So, so all we can say is so exciting, but we also have to throw up our hands and say there is a huge amount of complexity in this pathway that is regulating the timing between these regions. I know I don't have too much more time, and I want to try and wrap up in the next 10 minutes. So what we then did was say, okay, we've been looking under the street lamp. We've been saying, this is the pathway that is relevant, but it's also possible, since I said that this pathway isn't completely proven and there's still arguments about it, that we're not looking at everything, right? We're just looking at what we think is involved. So maybe we should do something more unbiased. And so what we did then was take this paper's description of 9 groups, or 9 clusters of Purkinje cell, and say, do these 9 clusters of everything that's in the transcriptome, do they align with the lobules? So this is now that same data plotted by lobule, and it's confusing. So I'm going to pull out all the other lobules and just show you the lobules that we're interested in. So that's 4, 5 in pink, flocculus out there, and 10. So based on their high dimensional clusters, again, the 3 lobules, they fall in different clusters. There is some overlap, but the 2 long delay regions also separate from each other. So if you were not to look just at the mLUR pathway, we really see that these regions are extremely different from each other, and really that really needs to be considered as well. So quickly to show you some more data, this is of course somebody else's data that we mined, and we were worried, we wanted to sort of validate this in our own hands to measure the RNA. And we were also interested in something that this RNA-seq data set does not have, which is the splice variants of the actual channels we're looking at, because metabotropic glutamate receptors come in different splice variants, in different isoforms, and so does the TRPC3 channel itself, which has not been very well understood. But in the mid-2010s, like about 2012, 2013, different splice variants of TRPC have been described. So we wondered whether it was the channel itself that was actually different. So we did RT-qPCR for different lobules and found that mGlu1B is different between 4, 5, and 10 in the flocculus. This is not unexpected because this actually goes with the general zebran identity of the lobule. That's what has previously been shown. But we also found that this short versus long correlated very nicely with a relatively recently described splice variant of the TRPC3 channel called TRPC3C. And without sort of belaboring the point of spending too much time on it, TRPC3C has different channel opening properties and lets in a different amount of current. In a way that explains the timing, no. But in a way that correlates with this timing, yes. So we also looked at, in addition to the RNA, we looked at the protein level. We ratioed the TRPC3 to the mGlu-R. You see differences across lobules in the direction that would be predicted by the RNA-seq data set. So all of that is nicely validated. And so we were left with that, with sort of this high level of complexity, but a suggestion for several pathway components that would all converge to act on shifting the timing and creating this heterogeneity across different synapses. So finally, the next thing that Ria was interested in doing that I want to tell you about is to wonder whether this slow synaptic current makes any difference to Purkinje cells at all, right? Because Purkinje cells, and this is from an old paper, which suggests, which shows that Purkinje cells in different lobules actually have different input-output relationships. So just injecting current into the cell does not necessarily result in exactly the same firing patterns across all Purkinje cells. And therefore, given that there is this complexity in input-output relationship, a depolarization does not always mean just an increase in firing, right? So how does the heterogeneity at the synaptic level interact with the heterogeneity of the cell's properties to ultimately impact the firing output of the Purkinje cell? So Ria sort of confirmed that you see the patterns of different cell types and the variability in firing rate that you expect across lobules. And then what she did was she first, in voltage clamp, characterized the slow EPSC, flipped to current clamp just below threshold for spiking, and saw when the spiking happened in response to that synaptic current. And as you can see, perhaps not surprisingly, the time of peak of the firing is really nicely aligned with the fast versus really slow dynamics across different lobules. And that this firing is true across a range of frequencies as well. So regardless of the Purkinje cell's intrinsic firing properties, it does turn out that if you give these stimuli to a parallel fiber, then what the Purkinje cell is outputting is also highly variant across timescales. So a parallel fiber input, essentially what we're saying is for lobules 4, 5, it's going to be active for like 300 milliseconds and shut off. It's going to have an increase in firing for that long. But in lobule 10, that can go out to over a second. The same parallel fiber stimulus would drive firing on this much longer time scale in the Purkinje cell. Now, of course, these are whole cell recordings, so we're artificially keeping the cell at a voltage, and Purkinje cells are naturally spiking cells. So this is again not the perfect experiment in a way, because what does it do to a naturally spiking cell that is spiking at its intrinsic firing frequency, which could already be quite high? Would this make a difference at all? So we did cell attached recordings where the cell was allowed to fire. We're not dialyzing the cell contents. There's no impact of our internal solutions on washout of any sort. And then we give the same parallel fiber stimulus, and you see a short, sharp increase compared to a much prolonged one in lobule 10 and the flocculus. We also repeated this experiment without any inhibition blocked, because inhibition is one of the regulators of Purkinje cell firing rate. And in all the previous experiments that I told you about, we had fast synaptic transmission blocked, both excitatory and inhibitory. Here you had no inhibitory block and no block of the AMPA receptor. You got a fast response because of the AMPA receptor's presence, but it shut off much faster in lobules 4, 5 than in lobule 10 and in the flocculus. And therefore, the same parallel fiber input is really being diversified across these different regions, driven by the slow synaptic current heterogeneity, which is driven by the heterogeneity in the mGlu TRPC signaling cascade that underlies it. So I'll try and wrap up there, because I want to finish at 45 and I'm 2 minutes over. I just want to acknowledge that the plasticity work started in Jennifer Raymond's lab. And this is my lab right now. Most of what I showed you was Ria's work. Some of it was Francisca's work. We collaborated with Keith Murray's lab, and we are funded by several people in Canada. So thank you for your attention. I'd be happy to take any questions.
That was marvelous, Aparna. Thank you so much. And if you have a question, please just go ahead and raise your hand, and I'll be happy to call on you. Let me start by, I guess, start the questions, Aparna. So if I wanted to get clarification, you showed that at the beginning of your lecture, the 120 millisecond delay in the climbing fiber input in the flocculus was effective in producing LTD. But same plots, I think, were showing that at 0 millisecond delay, if I understood it correctly, there was LTP. Is that correct?
Yes, that's absolutely correct. So when it was done synaptically, when we did a synaptic EPSP or EPSC recording, there was potentiation at 0 milliseconds. And this was particularly interesting because at these synapses, and we tested it both in the flocculus and in the vermis, if you gave the same stimulus pattern, so these stimuli are usually 300 times at 1 hertz paired. So if one gives exactly the same stimuli without the climbing fiber, so 300 times at 1 hertz, you drive non-associative potentiation of the parallel fiber, which is not something we've found that's been shown before by Kozmans and the Desio group. But we tested that this was indeed true. So what the floccular synapses were doing is they were doing exactly the same thing as if they didn't see a climbing fiber stimulus. So if you did the parallel fiber alone or you paired it coincidentally, it was just doing potentiation. And not if you had the delay. But in the vermis too, if you gave parallel fiber alone, you got this non-associative potentiation. Now, we did not go into molecular mechanisms of the potentiation versus the depression. So I couldn't really speak to that. Yeah. But it's a great point. Yeah, we really saw this increase rather than nothing when you do the synaptic recording.
And if I understood you correctly, you said that was equivalent to having no climbing fiber input.
Exactly. So if you gave the same 300 pulses at 1 hertz of just the parallel fiber, you would get the same potentiation.
Right. Yeah, wonderful. I wonder if you could speculate about why there might be climbing fiber input that appear to be not associated with any errors. So, for many tasks, many labs see climbing fiber activity near initiation of behavior. And yeah, having studied these timescales of learning, why are there complex spikes when you're just beginning to make a movement?
Yeah, this is an excellent question, but it's not one that I can answer. It's one that I can speculate. But part of it is that I feel like climbing fibers. So again, as a synaptic physiologist, we're putting in the climbing fiber as an error signal, as a teaching signal. But it's not that climbing fibers are not firing at 1 hertz all the time. So there is a signal that is. It's not like a reward signal that's coming in only when there's a reward or only when there's an error. There is something that the climbing fiber is signaling continuously. Now that firing rate goes up with errors, but there's also been evidence that it goes up with unexpected things, with rewards in different circuits, right? So I think the question really comes down to what all is the climbing fiber signal encoding, which I don't think we have a satisfactory answer to. And our kind of data is not getting to that, really, right? Because we're bypassing the question in a way of what the climbing fiber is actually carrying. We're just saying if it comes in, this is what it does.
Wonderful. I'll let Jennifer go ahead and please ask your question.
Hey, Aparna. So nice
Hi,
to see
Jennifer.
you, and congratulations on this work. You said that we have moved beyond the idea of universal circuit computation in the cerebellum. And why do you say that? Because I might think that the differences you're seeing at the cellular level are just sort of allowing the same computation to be done for behaviors that play out over different timescales. So do you really believe that? And what is your evidence?
I think you're right. I would say my reason for saying that was that okay, let's see. I think it's twofold. And perhaps you're right. I see your point that it's not that the entire computation is different, that we have evidence that there's a different computation in different regions of the cerebellum, right? But I think there is evidence that the cerebellum is not doing the same thing across regions just because of the diversity of behaviors it supports. That's one part of it. Just because some parts of it are doing sort of language and social behavior, and some are doing reflexive movements. Therefore, it's not doing exactly the same thing. That's one part of it. And the second part of why I said that it's more accepted it's not the same thing is because just from the cellular point of view, I think there are just so many labs showing that firing rates are different across lobules. Firing properties are different across lobules. Now we're saying synaptic properties are different across lobules, right? So the feeling that that wherever you looked in the cerebellum, you could just transfer from one region to the other is clearly no longer true, right? But I agree with you that maybe my saying that it's accepted that there is no universal, Schmann's universal cerebellar transform is no longer true is perhaps stretching it a little bit, because we don't actually know what that computation is in different regions and how it's different. Does that make sense? I think, Jennifer?
Thank you. Sasha, you have your hand up.
Thanks. That was beautiful work. It's like I sort of saw the paper, but I didn't read it, and so I appreciate your walking me through it. And
thank you.
You're welcome. So I'm just gonna. So I actually I had a question, but I want to just pick up on this discussion because when you said that it's generally accepted now that the computations aren't universal, I was like, if only.
That's true. That's true. I mean, in a way, this is exactly what we said in the paper. We were like, well, we think the parallel fibers are carrying the same thing, and it's going the same way through the circuit. And here is a piece of evidence against that. So I'm willing to step back on that statement and say that yes, you have a fair point. This isn't we can't accept that it's that heterogeneity of whatever computations the cerebellum is doing is widely accepted across the field, because
Right.
we
And
don't really know.
as
Yeah.
Jennifer just pointed out, and I think this is like all the young people in the audience and the old people who want to reboot, we don't know what the
We don't know.
computations are,
Yeah,
right?
we
That's the
don't.
fundamental. But from my perspective, thinking about timing and the nucleus neurons, and we've been diddling with the dentate, in which there are extremely teeny tiny neurons that nobody would ever patch from or record from, and huge neurons. And the timing signals are so radically different. I can't imagine the computations would be the same, but your work and others will show us. So my question is, I hate to like bring up human, but now I'm like, okay, so what is, are there, if you've looked at your gene expression data set and then what we know in primate human, you know, like, is there anything like qualitatively different? I don't know what I'm sort of asking, but like, do you think that we can get a real sense of the time scale diversity across species from the mouse, or do you think there's going to be some other thing in the human that's qualitatively different?
It's an excellent question, and one that I don't have an answer to, but it's one that we have been thinking about, not in terms of the human, but in terms of the primate,
Great.
as a sort of midway step. And again, I don't have an answer to this, because evolutionarily there's a lot that has changed in the cerebellum between the mouse and the primate. Even when we do it very simply, and I really don't know much, but when I try to talk to primate human systems neuroscientists about the lobules of the cerebellum, just from the point of view of trying to understand what might be the functions, you know, what do these different lobules support? Just that very simple question from a mouse person to somebody who works in rhesus macaques and humans. The anatomy of the cerebellum is so different already, right? Those lobules are subdivided so much more, for example, 4-5, for example, in the mouse versus in the human. It's just a completely different anatomy, just at that level. And from there into what they see with fMRI. So I've not been able to satisfactorily bridge that. The one thing that we are doing to try and get there, and this might take us a couple more years to do it, is that we sort of have collaboration set up where we're now getting marmoset data. So we're getting slices from marmosets whenever there's a death or something is to be euthanized. We sort of hang around there and we time it and we dash around, and the whole lab takes that one cerebellum and tries to get some recordings from it, to try and get a little idea about, you know, whether the simple questions, and what we're addressing is really simplifying the problem, right? We're looking at a small part of this puzzle, but just that small part of the puzzle. Is there, for example, this kind of heterogeneity in the primate as it is in the mouse? We have another project that looks at sort of endocannabinoid signaling, and we're trying to see that both in the mouse and the macaque and in the marmoset, but of course we're limited, so it'll take us a while to answer that question. And even then, our answers will not address what you're asking. Really, it'll address a piece of what you're asking.
Oh, I never ask questions that can actually be addressed. I just like to, but this is really exciting for the future. I love this marmoset mouse collaborative connection. Great, thank you.
Thank you, Aparna. And are there any other questions? Well, I hope you have a wonderful week. We're coming up to Christmas. This is our final talk of the year 2024. Aparna, I want to thank you for being the bookend for this, for our get-togethers, and I hope you have a wonderful holiday season, Christmas time and New Year, and look forward to seeing you all next year. Thank you.
Thank you so much. Thank you, everyone.